Hello, everyone, and thank you for joining today's SNEV talk. My name is Barbara, and I'm a PhD student at the University of Coimbra in Portugal, and I'm moderating this talk with Swagatam from the MSU Barodam, India. Now it's my pleasure to introduce our speaker, Dr. Nicole, a senior associated scientist at the National Institute on Aging, National Institutes of Health. Her work is focused on the molecular mechanisms of aging and age-related disease with a strong interdisciplinary approach, and her recent research explored the use of EVs as a non-invasive biomarker. Today, she will be speaking on innovations and challenge in extracellular vesicle biomarker discovery, and we are very excited to hear about her insights into this evolving field. Thank you for being today with us, Dr. Nicole. And for the audience, please feel free to submit your questions. And at the end, we can discuss a little bit more with Dr. Nicole.
Can everyone see my slides okay?
Yes.
Yeah.
Okay. All right, let me get the pointer up. All right. So first off, I want to thank SNEV for inviting me to give this talk in the Translational EV series. I'm very honored. And I'd also like to thank all the members of SNEV for all the work that you've been doing to connect EV research students globally. So today I'll talk, as mentioned, on innovations and challenges in extracellular vesicle biomarker discovery. And I'll also give a preview of some of our unpublished work to give you an example of how we're looking at extracellular vesicles as biomarkers of aging and age-related conditions. And so there's many challenges in extracellular vesicle biomarker discovery. I mean, one of the most challenging is the heterogeneity of extracellular vesicles. There's many different types of EVs and extracellular particles. And here in this cartoon over here on the right, I've highlighted several. The most popular and most well-known are exosomes. There's also microvesicles, exospheres, large oncosomes, and apoptotic bodies. So these different types of EVs and extracellular particles does prove to be a challenge in the field since they're derived from different biogenesis pathways. Biofluids contain a mix of extracellular vesicles from different cell and tissue types. And there's a unique profile of EVs in each bodily fluid. And so these also may have different cargo, in each body fluid. The properties may be different. For example, if you're doing enrichment for a different particular type of extracellular vesicle, this may differ in each different body fluid. The size can also be influenced by isolation method. And as you can see here, even in this little cartoon, there's a variety of different sizes of extracellular vesicles as well. And so I like to always say it's kind of like finding the needle in the haystack of finding the EVs within your body fluid of interest. For example, EVs make up a small percentage of bodily fluids. It's been estimated that it's 0.005% of blood, plasma, or urine. And so the complexity of bodily fluids, it's not one size fits all. So if you're looking at EVs in urine, your main challenge may be to separate your EVs from uromodulin, for example, or you have a large volume, so it's very dilute, your sample. Whereas if you're looking at, for example, blood plasma, there's a lot of serum proteins, a lot of lipoproteins, and that may be one of your main challenges. And again, so there's different challenges for each bodily fluid. And so I also wanted to highlight some of the resources for overcoming these challenges. So the International Society for Extracellular Vesicles, so ICIV, has task forces that have, some of which have focused on different body fluids. And so I wanted to highlight some of these resources that have been developed from these task forces that may help you if you're interested in a specific body fluid. So for example, blood, there's a blood EV task force. And there was a recent paper 2 years ago led by Fabrice Lucien looking at, that is focused on isolating EVs from blood. There's also a urine task force. And this paper was led by Ute Erdberger and, covers many of the different aspects of isolating EVs from urine. There's also a cerebral spinal fluid task force led by Ursula Sandu, a cell culture condition medium task force, a parasite task force, and a flow cytometry methods And this is the EV-FCM task force. This was led by Josh Wells. So I encourage you to look at these different resource documents, if you're interested in these body fluids of interest. And so here and with ICIV, I am a member at large, and I'm part of the Science and Journals Committee, where we oversee the Scientific Reproducibility Task Force and other aspects such as the journals. And so I just wanted to highlight on the ICIV website these different task forces as shown here, because I think this is important to know that if you're interested in looking at EVs as biomarkers or in these different bodily fluids, that there's different task forces that are focused that may help you, in your research. And so I encourage you, each of these are hyperlinked that leads you to the task force page that you can find more information, be involved, contact the task force leaders. They're always looking for people to be involved and who have interest. It may help because, it's not most of the these task forces are pretty active, and they're really trying to overcome some of these challenges in the field. So there's also challenges in preclinical variables. And here I'm just going to highlight some with blood, because that's we typically are looking at EVs and blood plasma in our laboratory. And so one of the a big difference is in biofluid collection. So there's different types of tubes that you can collect blood in for either plasma or serum, for example. There's different sites where you can collect blood, or if they're fasting. There's differences in handling and isolation protocols for how many times you spin, what centrifugal force, what temperature, for example. There's also differences in storage. And so these preclinical variables, some of which you may have control over, some of which you don't, because you may be getting your samples from, for example, a core lab. But it's important to know, this protocol. And there was a recent document that was developed by the Minimal Information to Enhance the Quality and Reproducibility of Blood Extracellular Vesicle Research, the MI Blood EV technical report. And what this report highlighted is the importance of just reporting your blood collection and processing parameters and variables. And this is because most often when we're writing our methods, especially for some of the journals that have like short word counts, you end up cutting some of these parts in the methods. And so often people don't report some of these preclinical variables, which may be very important. And so they developed this standardized reporting tool for blood EV research for humans, for example. And here you can just have little drop-down menus and add in your preclinical variables. It's not meant to be a document to standardize the field. It's meant to be a document to promote transparency in these preclinical variables, and to consider, when people are looking at different studies and how to compare. So I encourage you, if you're doing, for example, blood EV research, to use this standard tool and to have it as a supplementary figure within your manuscript. Other different challenges are differences in isolation methods and also in EV characterization methods. I'm just going to highlight here the isolation methods. So, when choosing an EV isolation method, it's important to balance both specificity and recovery. And I particularly like this snapshot of EV isolation methods that was from Clotilde Thierry's lab, and it was published in Cell 5 years ago. And here they have this PCA where you kind of can view the specificity versus recovery for the different EV isolation methods. So, for example, the oops, I think somebody's not muted. I think so. For the highest specificity, for example, immunoprecipitation or asymmetrics, flow field flow fractionation, or density gradient here. But the highest recovery but lowest specificity would be precipitation or filter concentration techniques. Here in the middle, that's highlighted, or size exclusion chromatography or differential ultracentrifugation. So it's important to consider both specificity and recovery when deciding on different EV isolation method, or what might be the best method for your research. And I also like the visualization of the different types of EVs by size and also the contaminants with proteins and aggregates, and how these different techniques can isolate these different populations or the different populations away from the different contaminants. And so I encourage you to look at this visualization, when deciding on what type of EV isolation method would be good for what you're doing. And it's also, again, it's not one size fits all, because how you decide on an isolation method is dependent on what is your downstream question and what do you want to look at, and so how deciding on these different types of methods, again, depend on your specificity and recovery and what your research question is. And there's many other considerations for looking at EVs for diagnostics or for biomarkers. And one of those is cohort design to include different populations. And here I'm going to highlight, a number of years ago, we wrote a review where we did a literature search to look at how EV characteristics were, in particular, concentration and size, cargo, or functional effects, and how they were influenced by age, sex, population demographics, and disease pathology. And so we focused the review on population demographics, age, and sex. And so I'm going to highlight some of the papers that we found that were looking at how human age affects EV characteristics. So for example, if we look at protein cargo here, there was, with human age, various proteins can go up and down, what's been reported. In particular, with human age, so as individuals get older, there's a decrease in apoptosis proteins in EVs. There decreased mitochondrial DNA and respiring mitochondrial DNA. But if you look in particular at saliva EVs with older age, there's a microRNA that is higher. There's CD151 and mucin 16 that also are increased. If you look at concentration and size, there's differences in EV concentration. And if you look in particular subpopulations of extracellular vesicles, many of them are lower with human age. And there's also a subset that are lower in urinary extracellular vesicles. There's differe's in functional effects. So if you have EVs from young or old individuals, you add them to cells and look at the cellular function. There's a higher level of colony formation capacity of hematopoietic stem cells with EV donor age. There's an increase in mitochondrial respiration with EVs from young individuals. There's an increase in EV internalization by B cells with EV donor age. And there's an increase in monocyte activation with EV donor age. So these are just some things to consider. And here I've just focused on the papers that we found that looked at human age in the context of extracellular vesicles. But I encourage you to look at, and definitely consider, the populations that you're looking at and how they may affect different extracellular vesicle characteristics. And why this is important is that there's a growing trend in using extracellular vesicles clinically. And so I really like this recent systematic review of extracellular vesicle clinical trials from A.Ju Wang and Randy Kearney. And what they reported was that there was a total of 471 clinical trials that were using extracellular vesicles. And if you look at the percentage, 47% were using EVs for diagnostic purposes, and 33% were using for companion diagnostics. So this means using EVs as a secondary outcome in the clinical trial. And in addition, there was 19% that were using them for therapeutic purposes. So you can see that probably almost 80% of the trials were using EVs for diagnostics and for biomarker studies. And so if we look more closely at these clinical trials and the isolation methods that were utilized, majority of the studies do not specify in their clinical trials how they're isolating extracellular vesicles. There was a small percentage, 12% that did. And so if you look at those 12% and look at the different isolation methods, the most popular was ultracentrifugation, followed by filtration and size exclusion chromatography. And so if you look at the different conditions and indications with EVs that are focused on, EV clinical trials focused on diagnostics, the top condition was lung cancer, followed by prostate cancer, followed by pancreatic cancer. And so the top indication category was then cancer, and then followed by respiratory, neurological, and cardiovascular. If we look at the top EV source, it was blood, followed by urine, and then plasma and serum. So here they've separated the categories, based on whether they indicated blood or partitions, particularly in serum or plasma. So if you combine these categories, the top EV source with blood would be much larger. So this is looking at EVs diagnostics. So if you look at this in the EV clinical trials focused on companion diagnostics, so again, this is using EVs in the context of a secondary outcome. The top condition was obesity, and this was followed by lung cancer and breast cancer. And then if you look at the top indication, it was by far cancer, followed by neurological, and then cardiovascular, and then obesity. And then if you look at the top source, the top source was blood, followed by plasma, followed by urine, followed by serum. So this kind of just gives you an overview of the interest and what types of clinical trials are utilizing EVs for diagnostics or companion diagnostics. And this growing trend in the field is really being pushed by innovations in the extracellular vesicle field and in utilizing extracellular vesicles in various clinical trials. And so I just wanted to highlight some of the innovations in extracellular vesicle biomarker discovery. And again, this list is not exhaustive, but just to highlight some that are really pushing the field forward. And these include multi-omic technologies, tissue biofluid extraction and analysis, AI-based machine learning tools, single EV technologies, focusing on imaging, capture, cargo analysis, for example. There's been innovations in lab-on-chip devices, high-resolution flow cytometry, microfluidics, and also acoustic fluidics. And again, I just wanted to kind of highlight some of these different innovations, but again, this list is not necessarily exhaustive, but just to give you an idea of some advances in the field lately. And I do think it's important to highlight that these innovations are sparked through discovery of and collaborations between scientists of different disciplines, for example, bioengineers, basic biologists, biophysics, and so it's important, in your career, especially with you guys being early career scientists, if you see a problem in the field, it's important to reach out and, reach out to an engineer, for example, if you're having trouble with and you think there would be a good technology that might help you in your studies. And so I encourage you throughout your career to have a multidisciplinary approach, and then also to reach out again to other scientists in different disciplines. And so now I'm going to switch gears a little bit and talk a little about how we're utilizing extracellular vesicles as biomarkers of aging and age-related conditions. And I'll highlight some of the work we've done, and then I'll talk a little bit about an ongoing research project in the lab that's unpublished. So a number of years ago, we were interested in extracellular vesicles in the context of human aging, and we found that with advanced human age, we see a decrease in circulating EV concentration. And we looked at the mechanism, and this mechanism was driven in part by an increase in the internalization of EVs from old individuals by B cells compared to EVs from young individuals. And we followed up, and in this work... Okay, can you hear me now? Okay. And so then in this work, we also looked at extracellular vesicles and some of the protein cargo, and we followed up this work looking at mitochondrial DNA as a cargo in extracellular vesicles. And I'll talk a little bit more about why we're interested in this and a little bit later in the talk. But we found that with advanced age, there's a decrease in EV mitochondrial DNA. We've also looked at demographic differences in EV protein cargo. And then if we look at EV protein cargo and characteristics, they were associated with clinical markers of mortality. So that led us to a follow-up project where we looked at EVs and mortality. And we found that there were higher EV-associated inflammatory proteins with mortality. We've had a number of papers looking at EVs in the context of diabetes. So with the acquisition of diabetes, there's higher circulating levels of extracellular vesicles. There's also different protein cargo and inflammatory proteins, and in particular, VEGF-A in extracellular vesicles. And VEGF-A levels correlate with disease severity. And so here I'm going to talk now about some unpublished work in the laboratory where we've been looking at chronic kidney disease and extracellular vesicles in the context of chronic kidney disease. So in the US, CKD affects about 35 million adults, and it's an important age-associated chronic disease. And if you look here at this percentage of adults, it increases with age, and then 35%, 34% of adults 65 or older have CKD. And in particular, we're also interested in obesity. So approximately 42% of adults in the US are obese, but we don't fully understand how obesity contributes to CKD. Obese patients progress to end-stage renal disease faster than non-obese patients, and the rate of end-stage renal disease increases directly proportionate to body mass index. So obesity is also associated with more proteinuria and enlarged glomeruli, which result in downstream health consequences such as renal insufficiency, hypertension, and obstructive sleep apnea. And so the connection between obesity and CKD is especially important not only here in the US, but worldwide, given the rise in obesity rates worldwide. So 43% of adults are overweight and 16% are obese. And so here we want to ask the question, are EVs the link between obesity and CKD? And so we identified participants in the Handles cohort who at visit 1 were obese without CKD. And then over a 5-year period, they developed chronic kidney disease. And this was a longitudinal cohort, so we have samples both at visit 1 and visit 2. And we matched these CKD obese participants with obese participants without CKD. And we had 3 controls to 1 CKD participant. And we matched them based on race, sex, poverty, and age. And so we have this longitudinal cohort of participants in Handles who, again, at visit 1, did not have CKD, and a subset who acquired CKD over a 5-year time period. And so our experimental workflow was that we had participants, again, with or without CKD over a 5-year time period. We had blood plasma. We had 135 samples because we had them at both visit 1 and visit 2. And then we isolated our EVs using size exclusion chromatography. We combined the fractions 1 through 3 for the EV enriched fraction. We isolated DNA and performed qPCR looking at mitochondrial DNA, and I'll talk a little bit more in detail about this. We also characterized the EVs using nanoparticle tracking analysis, Western blot, and also electron microscopy. And so here I'll just show our characterization of EVs, and we did this according to MISIV guidelines from ISIV, where we have the presence of known EV markers, CD9 and flotilin-1 in our EV enriched fractions 1 through 3. And we have the absence of the purity marker GM130. We've also done albumin assays, ELISA assays, to show that albumin elutes later in the later fractions. And here we've have an electron microscopy image, and these show the typical morphology of extracellular vesicles and the size from plasma. And here you can see one EV, for example, that has the cup-like morphology that often occurs during the dehydration process of electron microscopy. On the right here, we have size distribution graphs from nanoparticle tracking analysis. And so here we have all the EVs that were isolated from the average of the EVs from visit 1, and then here's from visit 2. And these have the typical size distribution of EVs that are isolated from plasma. And so our EVs that we isolated using SEC for this EV enriched fraction has the characteristics that are typical of EVs, so the protein markers and the size and morphology. And so next we wanted to look at, is EV mitochondrial DNA cargo associated with CKD status? And so why are we really interested in mitochondrial DNA as cargo in extracellular vesicles? So mitochondrial DNA can be released outside of the cell as circulating cell-free mitochondrial DNA. And here's just kind of a little cartoon of this. And it can be reliably measured in serum and plasma, and typically just in whole serum or plasma is it examined. And here it can act as a damage-associated molecular pattern molecule following cellular damage or stress. And we found that circulating cell-free mitochondrial DNA is present in plasma EVs under normal physiological conditions, and that plasma EV mitochondrial DNA levels decline with human age. And this is important since we know that mitochondrial dysfunction occurs with human age. There's been a really emerging interest in looking at mitochondrial components in extracellular vesicles. And there's several papers that have looked and can visualize mitochondrial components in EVs. And here I've just highlighted this one electron micrograph from, and this was from a report that looked at mesenchymal stem cells that were stimulated. And you can see a whole mitochondria and even individual cristae within this extracellular vesicle. Efrat Levy's lab has reported that EVs from the brain contain a population of mitochondrial-derived vesicles termed mitovesicles. And so there's definitely a emerging interest in looking at mitochondrial components within extracellular vesicles. And so we developed a pipeline for quantifying mitochondrial DNA from extracellular vesicles. So we have our plasma, we separate them using SEC. We have fractions 1 through 3, and then we DNA treat the extracellular vesicles to remove any DNA that's on the outside of the extracellular vesicles or that may have co-precipitated during the isolation process. We then isolate the DNA, and then we perform qPCR, and we do this with a slightly different approach in that we use different primer probe sets against 4 different regions of the mitochondrial genome, as highlighted here in these red boxes. And here we've named these using the starting nucleotide to each of the different primer sets. So again, we're amplifying 4 different regions of the mitochondrial genome, as shown here. So we've isolated the DNA, we've isolated the EVs, and then DNA is treated, and then isolated the DNA on all 135 samples within our CKD cohort, and then have looked at the mitochondrial DNA levels with all 4 of these different primer sets. So we first looked at the relationship between the different primer sets, and they're all positively correlated. And so if we look at, for example, here, 3164 compared to 4625, mitochondrial DNA levels are very significantly positively correlated. And this is consistent between all of the different combinations of the different 4 primer sets. So we see a positive correlation between EV mitochondrial DNA levels. And so what about, is EV mitochondrial DNA cargo associated with CKD status? So we first did a cross-sectional analysis at visit 1. So just to remind you, visit 1 is when both the controls and the CKD group are without CKD. So they're obese without CKD. And this is cross-sectional, and we've done linear regression modeling to the study design, so including the variables race, poverty, sex, and CKD status. And so here, the group that will later develop CKD, is labeled here CKD in lavender, and then we have navy for controls. And so African Americans who will later develop CKD have lower EV mitochondrial DNA levels compared to African American controls, and then also compared to white participants who will later develop CKD or controls. And if we look at poverty status, we saw an interaction with poverty, whereas those living above poverty who will later develop CKD have lower EV mitochondrial DNA levels compared to above poverty controls or below poverty participants. And then we also saw an interaction with sex, and that men who will later develop CKD have lower EV mitochondrial DNA levels compared to control men or women with or without later development of CKD. So again, this is data at visit 1 and then a cross-sectional analysis. And so we're also interested in the relationship between mitochondrial DNA and ancestry in this cohort. And so we examine ancestry looking at mitochondrial DNA haplogroups. So mutations in mitochondrial DNA amass through maternal lineage, and this allows for specific single nucleotide polymorphism combinations to be identified. And these SNPs can then be traced through ancestry, and the SNP combinations delineate a mitochondrial DNA haplogroup, as shown here. And this is important because in vitro studies have found that mitochondrial DNA haplogroup can affect mitochondrial function. So what we did was we haplotyped the participants in our cohort, and then we assigned a haplogroup for each participant. And if you look at the top level haplogroups, in general, there's three major: European, Asian, and African. And in our cohort, we have majority are European or African haplogroups. And so we are also interested in looking at the relationship between EV mitochondrial DNA and ancestry, because we've found in our previous manuscript that extracellular vesicle mitochondrial DNA levels are associated with race and mitochondrial DNA haplogroup. And this is work by two talented postdocs in the laboratory, Anjali and Victor, who are now at Ohio State and Johns Hopkins for graduate school. And what they found was that African-American participants with the African mitochondrial DNA haplogroup had higher levels of EV mitochondrial DNA, and there were also differences in circulating inflammatory protein levels with both race and haplogroup. And so this spurred our interest in looking at haplogroup initially in our cross-sectional analysis. And consistent among all three of these different primer regions, what we found is that those with the African haplogroup who will later develop CKD have the lowest level of EV mitochondrial DNA compared to African haplogroup controls or European haplogroup who will later develop CKD or controls. And again, this was consistent with data from three different regions of the mitochondrial genome. And so mitochondrial haplogroup and CKD group are associated with EV mitochondrial DNA levels. And so just to kind of reiterate that next we're going to look at, we're going to do a longitudinal analysis. So we have samples and have looked at EVs and EV mitochondrial DNA level at both visit 1 and at visit 2. And so now we're going to do our longitudinal analysis, looking how the development of CKD affects extracellular vesicles and extracellular vesicle mitochondrial DNA level. So in our longitudinal analysis, what we found was that EV mitochondrial DNA levels are lower in participants who developed CKD compared to controls. So this again was in our longitudinal analysis, and we found that individuals who develop CKD have lower EV mitochondrial DNA levels. So we were interested in, does this happen in both those with the African haplogroup and the European haplogroup, or in just one of the populations? So in order to do this, we did a stratified analysis by haplogroup. And the stratified analysis, in this stratified analysis, what we found was that only in those with the African haplogroup are there lower EV mitochondrial DNA levels for those who develop CKD. This was not the occurrence in Europe, those with the European haplogroup. We also found that with advancing age, there was an increase in EV mitochondrial DNA levels in those with the African haplogroup. And that if we look at sex, women have higher EV mitochondrial DNA levels versus men, but only those in the European haplogroup. So in our longitudinal analysis, we found that EV mitochondrial DNA levels depend on mitochondrial DNA haplogroup for CKD status, age, and sex. And so just to summarize this part of my talk, so what we found was that in African American and white obese men and women who acquired CKD over a 5-year time period, those with the African haplogroup had lower EV mitochondrial DNA levels with CKD. And so what does this mean in the context of data in the literature? And what can we glean from these findings? And so it's known that lower circulating cell-free mitochondrial DNA levels were reported in patients with renal dysfunction, including CKD. But these papers were looking at whole serum or whole blood. So whole blood would also include, and the majority of the mitochondrial DNA would then be coming from peripheral blood mononuclear cells. Levels of mitochondrial DNA are directly correlated with energy reserves, oxidative stress, and changes in mitochondrial membrane potential. So your levels of mitochondrial DNA determine, the energy reserves. Lower circulating cell-free mitochondrial DNA number is associated with mortality and age-related disease. So if you have less mitochondrial DNA copy number, you're more likely to have mortality or age-associated disease. And circulating cell-free mitochondrial DNA copy number has been proposed as an indicator of renal mitochondrial function. So what we think how this may relate to our findings is that lower EV mitochondrial DNA levels with CKD may be an indication that there's reduced renal function, in these participants. So it may be a biomarker of renal function or renal dysfunction in this case. And so here, hopefully, I've giving you some a gleam of the work that we're doing and looking at extracellular vesicles of biomarkers of aging and age-related conditions. And so we've looked at EVs in the context of aging, also in diabetes, mitochondrial DNA haplogroup, and there's the unpublished work that I just spoke about as well. We've looked at EVs in the context of mortality and also frailty, which I didn't have time to mention today. So here, just how we're, kind of the thought process on how we're looking at EVs in aging and age-related conditions. And then I'd also wanted to bring up, for those of you in SNEV, that there's, ICIV has, next year will be in North America. And so ICIV is going to, ICIV 2026 is in Puerto Rico next April. And that ASIC, so the American Society for Intracellular Communication, has a meeting in Potomac, Maryland in October. I also wanted to highlight the ICIV SNEV mentorship program that I'm involved in as well. And so just wanted to highlight some different conferences upcoming that may be of interest to you. So I am a senior associate scientist in Dr. Michelle Evans' laboratory as part of the Laboratory of Epidemiology and Population Sciences. So would especially like to thank her. The EV-CKD work was primarily done by a very talented postbac, Jada Morgan, who's now at Columbia University in medical school on a free ride there. I also work very closely with our statistician, Nicole Mode, and also another statistician, Alan Zonderman. I'd also like to thank, so we have a current postbac, Brittany Black, as shown here in Althoff Lahoney, and then also the handle study participants and staff. And I'd be happy to answer any questions now, or you can feel free to email me or find me on social media or LinkedIn. And I always like to kind of end a little early to make sure that I have plenty of time for questions and discussion. So I'm happy to answer anything now.
Fantastic talk, Dr. Nicole. It was very insightful and quite interesting to see a longitudinal study of EVs. So I hope there are some questions coming up. Probably our participants would want to ask some. But in the meantime, I do have a question that I'd probably start off the ball with. So did you see any cohorts that you studied with? Were you also noting that comorbidities apart from CKD that you were looking at and obesity? Were any other also recorded? And if so, which were the ones that you usually saw quite common?
You mean as far as, so did we look at comorbidities within the CKD cohort?
Yes, yeah.
So no, in general, we didn't. We did exclude people with diabetes in that cohort, since diabetic renal disease, may be a different etiology than chronic kidney disease. So we did exclude participants with CKD. But because it's a longitudinal study, it's hard to find participants that fall into that different category. So we can't look at everything, because then our degrees of freedom for our statistical analysis goes lower and lower. So in general, we did not look at other comorbidities in this specific project.
Okay, and one last question. So given the maternal inheritance of the mitochondrial DNA, so how do these haplogroup studies complement the nuclear genome analysis, if you have any insight on that?
Yeah, so we didn't do any nuclear genome analysis in this cohort.
Okay.
We're deriving ancestry from haplotyping or haplogrouping, our participants. It's something that we're interested in, is looking at whole genome sequencing in this cohort, and we do have some data. We just haven't had a chance to look at that, but it is something that we're interested in.
Great. Okay. So I think we can take some questions. So there's one question that says, what is your opinion regarding tangent flow filtration as EV's purification method?
Oh, I mean, sometimes with the technologies, I am not a technology-driven person. As you can tell, I'm a basic scientist. So I wouldn't say I can give a strong opinion on it. I've never used it myself, so, or in our laboratory. So, I mean, it seems to be a growing trend to use it. So I would say that, but I can't really give any sort of personal recommendations on it or not.
Right. And there's one that's asking if you check the mitochondrial DNA inside the small or the large EVs.
In this project in general, we didn't differentiate between small and large. We just combined them. So they were the EVs that were isolated using SEC, which contains probably a mixed population. We didn't differentiate based on size.
Okay. So, and do you know if the, sorry, yeah, go ahead.
If I can ask, Dr. Hooten, thank you. Excellent talk. For the first part, I had two questions. For the first part, just largely as EVs as a biomarker, I was happy that you mentioned Dr. Carney's review. It's interesting to me, and I wanted to gauge your thoughts as to why you feel like EVs seem to not have taken off in the clinical space, especially when compared to some of its other liquid biopsy counterparts, CT or CF DNA. If you could speak to that, it seems like it's largely still in the preclinical realm, and it hasn't seemed to drive clinical decision making much.
I think there's probably a number of different obstacles, one being the variations in the different isolation techniques. So you can imagine, in a clinical space, if you're trying to do ultracentrifugation, because that seemed to be the most popular one, that's a very technical, approach that you can't do. Some of the biomarker studies, are very high throughput. If you're looking at, CRP, GDF15 as a biomarker, you can do those pretty high throughput. EVs, have not been necessarily, I think that's we're at the cusp of some of the technology to really push forward some of the high throughput, and I think that's where we're limited, and I actually did have a slide, but I decided not to do it. I mean, I think that's one of the barriers in the field is that, you have biomarker studies on this side. So if your paper goes to a biomarker person,
Sure.
They're like, why can't you do it in 1,000 people? And you're like, that's really hard. These things take time. And, and then, so trying to, and so I think that's part of the issue is or CLIA lab, all those different parameters are really, I think we're still, like you mentioned, in more the preclinical phase because of that technology that someone can do in a clinical lab.
Sure. And do you think that any of that ctDNA or cfDNA that we are measuring in clinic, do you think any of that is also measuring EV signatures as well?
Probably, because it doesn't differentiate, right? I mean, when you're doing and when you're doing circulating DNA in general from serum or plasma, you're not differentiating whether it's from EVs or from,
Sure.
The general plasma or not.
And then, sorry, last question about the second part about your CKD. If I can ask, did you find that, if I get this correct, the EV mitochondrial DNA inversely correlates with kidney function? Were you able to find that was true across CKD stages 1 to failure?
Yeah, we didn't look at that because, this is kind of our, first endeavor into the CKD
Sure.
space, looking at these participants. And, Handles which I didn't go as much detail in this talk because it was, I was focused on other things. Handles is an observational study. It's not
Sure.
a clinical study. So we know over this time period, they didn't have CKD, they do have CKD. I think those type of studies would be better, in, a more clinically focused patient population. These are participants. And then also, if there is some, one of the studies that looked at circulating cell-free mitochondrial DNA from serum, so not from, EVs in general, they find it did correlate with some of the stages of renal disease.
Interesting. Okay. Thank you.
Thanks.
Okay, so we have a couple more questions. I'm going to quickly go through them. So do you know if the mitochondria in EVs are functional in control and dysfunctional in CKD?
Ooh, I mean, yes. So we have not measured their function in this assay. That would be very good to do, very interesting to do. We can also, add them to cells and look at mitochondrial function, but I mean, that would be something that was interesting to do, but we have not done functional assays, in
Have
this
we,
study so far.
okay. Have you also looked into mitochondrial damage?
Mitochondrial damage, we have not looked at, no.
Okay. And there's one question about relationship between EV size and carrying cargoes exist. Is that something that we can focus on?
I think there's in general, people think so, because of the physical properties of, you can only fit so much in a small EV versus a large EV. I think that's most of the, general in the field. It's thought that there'd be more cargo or different cargo. I mean, proteins are and microRNAs and all these are different sizes. So, your cargo may vary, in EV size and because I think the size of the EV sometimes, not always, denotes the biogenesis pathway too. So it may be that, if you have a large oncosome that's from a cancer cell that may have, a lot of cargo that's different than some of the small EVs.
Right. Also, if we want to start off by just seeing what kind of EV population we get, irrespective of any size range or anything specific we're looking for, what methods would you recommend for one to go ahead with if the biofluid is, let's say, blood?
I mean, we've been utilizing size exclusion chromatography in the lab for a number of years and if you use, I don't want to promote specific companies, but we use the automated fraction collector from Izion, and that seems to work pretty good as far as reproducibility, within measurements, depending on the different columns. Some of them have better specificity than others, or sometimes they're more dilute. So you may have to concentrate your samples before or after, depending on what type of body fluid you're using. But we've been using that lately in the lab, and it's been successful, and it's not super, it's not super labor intensive. I mean, it is because you're kind of waiting. You're sitting there, waiting for the fractions to go, but it's not, like some of the other techniques that take, 5 hours to do 3 samples, and you're afraid you're going to blow up the, centrifuge or something if it's not balanced,
Right. We do have one question on if you could elaborate a little bit more on the ISAF mentorship program.
Okay, so ISAF it's a pilot. So they just started after the ISAF tech meeting. So it was probably like in December, they asked for applications for ISAF SNEV mentorship program. So, for myself as a mentor, I provided an application, on why I wanted to be a mentor. I think the same was done on the SNEV level for students, and then they matched you to a student. They didn't do, this was more meant to be, and they started small with a smaller cohort of ISAV and SNEV mentorship program because they didn't want to overwhelm. They wanted to make sure it was going to be successful before they kind of did it in a large group. And so with that mentorship program, we meet, my mentee and I meet roughly once a month, but it's kind of up to you on how often you meet to talk about, it's meant to be someone else in the field to give you advice on, guidance on career and how you're doing in your whatever type of career stage you're at, maybe some technical advice, but it's not meant to be super technical that you're giving experimental advice, but sometimes that does occur too if there's questions. So, sometime there'll be another data call for it, data call for a next cohort. So you can kind of look out for that in your, SNEV network emails.
Right. We do have a couple more questions, so I'm going to go through that as well. Why do you think the mitochondrial DNA will reduce and not be increased due to cell stress or damage?
Yeah, so, this is where we were gleaming data from the literature, because sometimes you would think it would be increased, because we do see an increase, like, for example, with frailty. But what we do think is that, data says that with mitochondrial function, and when there's reduced mitochondrial function, with lower mitochondrial DNA in the kidneys, for example. So this may be reflective of lower EV mitochondrial DNA levels being lower may be reflective of lower renal function And that lower renal function is due to lower mitochondrial DNA copy number within the renal tissue.
Right. And one question about whether you reuse the SSE columns or do you not do that to risk contamination?
Yes, we reuse them. Yeah, I mean, because everything right now is really expensive. So we've done it according to guidelines from the manufacturer. I think now we wash with sodium hydroxide, I think,
Right.
if I remember correctly.
I think you
But
can
we do
reuse that for like 2 to 3 times. I think that's enough, I
No,
guess.
I think we can. I think we do more than that. I think it's recommended 5, maybe. I have
Okay.
to look. But we whatever manufacturer recommends is what we do.
Right. So another question is, do you think EV biogenesis could be changed by age in CKD or more should be to kidney dysfunction?
Yes, I do think that and one of the things that we did find, at least for circulating EVs, we did not find a difference in concentration. I didn't really mention that or because of the shortness of the talk, but we didn't find any difference with concentration, in our cross-sectional or longitudinal analysis. So it doesn't, but that's again, circulating EV concentration, which could be, the EVs from a number of different sources, but here we do think it's reflective of kidney. And we do think, with human age, we did find that there was lower EV concentration with human age, and that there's lower EV mitochondrial DNA levels with human age, which may be reflective of, we know that mitochondria dysfunction is a hallmark of aging, and that mitochondrial function decreases with aging and contributes to aging and many age-related diseases.
Right. Okay. And any idea why people are analyzing in majority plasma-derived EVs and not serum-derived EVs?
I think people use both, and some of that's limited on what they can get, like if you're only, I think when we first started using plasma and found that there were more EVs in plasma than serum, that was just anecdotal. We don't have anything published on that. And there's a bunch of different papers that have looked at some of those preclinical variables and how they affect EVs. We've just consistently used plasma over the years.
Right. And is there any way to look into mitochondrial DNA transfer between cells? And which EV column would you recommend for best DNA RNA yield?
Okay, so the first question, yes, mitochondrial transfer is very big in the field right now, especially in cancer cells. So it's a way for cancer cells to affect lymphocytes. There's been a couple of papers. There was actually one that came out yesterday, I think, in Cancer Cell that looked at mitochondrial transfer. So that is a very emerging topic in the field, and especially if EVs are that culprit that is transferring EVs from different cell types. And so we haven't done that necessarily in our studies, but I do think that is a really emerging, very hot topic in the field. And what was the last question? I mean, the issue, with some of the SEC is just the amount of material that you get. And so I think it depends on what your upstream, you're looking at blood, you're looking at urine, and then, the SEC column for RNA, we haven't used it in particular for RNA, so I can't really speak on which one I would recommend. I think it just depends. Again, how much do you worry about, your specificity versus your recovery? Because in RNA, you really need more material, especially if you're going to do RNA-seq. So that's important to kind of consider, I think, the amount of material that you're getting, from your isolation technique.
Right. Okay. And how do you concentrate the EVs after SEC method by ultracentrifugation?
We have not used ultracentrifugation after we've concentrated using Amicon filters. So we've done that actually in other types of experiments, not necessarily in this project looking at blood, but we've concentrated the EVs using Amicon to be able to put on the SEC column if we've used conditioned media. We've also used Amicon filters on the downstream. When we've concentrated them to look at sometimes the protein cargo, like for example, Western blotting, it takes a lot of material. So we've taken a subset of that fractions 1 through 3, and we'll take a subset and then of that, and we'll kind of partition it out for different assays that we want to do. But part of that we partition to concentrate to lyse directly to be able to use for protein analysis.
Okay. And did you see a correlation with BMI specifically? Were there any differences with different sex?
Okay, so in this cohort, we did not, we matched based on BMI. So that was not a variable you can look at because they're matched. and I should have mentioned that because they're all obese. But that's something that we're interested in, is looking at, how does BMI, differences in BMI with CKD as well. But
Would
that's something that we're
having another I think addition of exercise versus people who don't exercise, would that also be a good variable in this case? Because in many papers they've mentioned that exercise does increase EV numbers, and
Yeah, I mean, exercise data is this is an observational study. So, you're kind of asking questionnaires about exercise data. We have actually a subset of people who do have accelerometer data, but that's really difficult to look at, and in a subset of our participants, it's something that I think is definitely important. And to answer the BMI question, other studies have looked at BMI and have found that BMI does increase circulating EV concentration. And when we've looked at EVs in the context of diabetes, we also have, I'm trying to think, no, in most of those studies, we actually matched for BMI because of some of the data in the literature suggesting that it did affect at least circulating levels.
Right. Okay. And I think this would be the last question for the talk. Is there a comprehensive review that categorizes blood plasma particles considered as contaminants sorted by the type of downstream analysis they interfere with?
Is there a what was the first part? Sorry.
Is there a comprehensive review that categorizes blood plasma particles considered as contaminants sorted by the type of downstream analysis they interfere with? I think that's a very specific.
So if you're looking for manuscripts to help you, I would definitely look at the Blood EV Task Force paper from Fabrice Lucien. I'd also look at MyCiv. MyCiv has a paragraph on plasma and what are some considerations and what are some parameters that you should look at when characterizing EVs, or what are important contaminants to consider or to report.
Right. Okay, I think that's all for today. There were a lot of questions, and thank you for answering all of them. And on behalf of SNEV, it was lovely to have you. And and I hope all our participants had a good session as well.
Yes, and for all you guys, just feel free to email me or follow up if you have additional questions, or if you want any. I think there were some methodological questions, so if you want any more details on protocols, you can feel free to email me. Thank you for having me.
Great.
Thank you so much.